Hello students, in this video we will do quick revision of facts and data based information from chapters 2 to 6. These chapters are going to come in your first term exams. First, bacteriophage known as 5174 has 5174 is a single stranded DNA virus which attack E. coli and it has 5386 nucleotides. Second, bacteriophage lambda has 48,502 base pairs and it is a bacterial virus that also infects E. coli. Third, Escherichia coli has 4.6 multiplied by 10 to the power 6 base pairs. Fourth, haploid content of human DNA has 3.3 multiplied by 10 to the power 9 base pairs, while diploid content of human DNA has 6.6 multiplied by 10 to the power 9 base pairs. Point number 5. A typical nucleosome contains 200 base pairs of DNA helix. Nucleosome is the structure formed when negatively charged DNA is wrapped around positively charged histone octamer. Point 6. The distance between two consecutive base pairs in DNA molecule is equal to 0.34 nanometer, or you can say it is equal to 0.34 multiplied by 10 to the power minus 9 meters. 7th, the length of DNA double helix in a typical mammalian cell. As a typical mammalian cell is diploid, so here you will take the value of base pairs as 6.6 multiplied by 10 to the power 9 and then multiply this value with the distance between two consecutive base pairs in meters, which is equal to 0.34 multiplied by 10 to the power minus 9. After multiplying these two values, the length of DNA double helix will come out to be 2 meters. यहाँ multiplication को देखिए, 10 to the power 9 and 10 to the power minus 9 cancel हो जाएंगे and by multiplying 6.6 with 0.34 we will get the answer 2 meters. 8th point, time taken by E. coli with 4.6 multiplied by 10 to the power 6 base pairs to complete the process of replication is 18 minutes. E. coli takes 18 minutes to complete the process of replication. Point 9. Number of codons for 20 amino acids. 61 codons are responsible for coding 20 amino acids. Point 10. Number of stop codons. There are 3 stop codons, UAA, UAG and UGA. They do not code for any amino acid. So, in total there are 64 codons. Point 11. The average gene consists of 3000 bases, but sizes vary greatly. Come to point 12. Largest human gene, dystrophin, it has how many bases? It has got 2.4 million bases. Point 13. Percentage of nucleotide bases which are exactly same in all people. It is 99.9%, means almost all, that is 99.9% of nucleotide bases are exactly same in all people. Point 14. Percentage of genome code for proteins, it is less than 2% of genome that codes for proteins. Point 15. Number of genes on chromosome 1 is 2968. Chromosome 1 has the most genes and number of genes on Y chromosome is 231. Y chromosome has the fewest number of genes. Point 16. Number of locations identified by scientists in humans where single base DNA differences occur. Scientists have identified 1.4 million locations where single base DNA differences occur in humans. Point 17. Adenylate residues added at 3' end during tailing process for processing heterogeneous RNA. 200 to 300 adenylate residues are added at 3' end of heterogeneous RNA during its processing, and the adding of adenylate residues is called as tailing. Point 18. Number of chromosomes present in a person with Turner syndrome. 45 chromosomes are present in a person having Turner syndrome. This is one less than normal person with XO condition. Point 19. Number of chromosomes present in a person with Klinefelter syndrome. 47 chromosomes, that is one chromosome more than normal person present in a person with Klinefelter syndrome with XXY condition. Point 20. Number of chromosomes in a person with Down syndrome. Here the number of chromosomes are 47. 47 chromosomes are present with trisomy of 21st chromosome in a person with Down syndrome. 21st. The monohybrid phenotypic ratio is 3:1, and the monohybrid genotypic ratio is 1:2:1. 22. Dihybrid phenotypic ratio is 9:3:3:1, and dihybrid genotypic ratio is 1:2:1, 2:4:2, 1:2:1. Point 23. Test cross is a cross between. It is a cross between capital T small t and small t small t. That is, it is a cross between F1 generation and its pure recessive parent. Point 24. In incomplete dominance, both phenotypic and genotypic ratios are 1:2:1 in case of incomplete dominance. 25. The embryo with 8 to 16 blastomeres is called as morula. Point 26. The number of primary follicles left in each ovary at the time of puberty. 60,000 to 80,000 primary follicles are left in each ovary at the time of puberty, because from birth to puberty large number of primary follicles degenerate. 27. The length of each human ovary is 2 to 4 centimeter, and each fallopian tube is about 10 to 12 centimeter long. 28. Number of compartments, also called testicular lobules, in each testis are 250. Testicular lobules are present in each testis. Point 29. The seed of lupinus germinated after an estimated record of 10,000 years of dormancy. The seed of lupinus was excavated from Arctic tundra and germinated and flowered after an estimated record of 10,000 years of dormancy. Point 30. 2,000 years old viable seed is of Phoenix dactylifera, which was discovered during the archaeological excavation of King Herod's palace near the Dead Sea. 31. Number of pollen grains formed from one microspore mother cell is 4. Microspore mother cell undergoes meiosis and form 4 microspores which develop into 4 pollen grains. 32. Number of embryo sac formed from one megaspore mother cell It is 1. Megaspore mother cell also undergoes meiosis and form 4 megaspores, out of which 3 gets degenerated and 1 develops into embryo sac. 33. Each embryo sac has 8 nuclei and 7 cells. 3 are antipodal cells, 2 synergids, 1 egg cell and 1 central cell with 2 nuclei. 34. Ploidy level of nucellus, endosperm, polar nuclei, megaspore mother cell, female gametophyte respectively are: nucellus is 2n, endosperm 3n, polar nuclei n, megaspore mother cell 2n and female gametophyte is n. Point 35. How many types of gametes are produced by individuals with genotypes? कितने टाइप्स के गेमीट्स प्रोड्यूस होंगे फॉर अ गिवन जीनोटाइप? इसको कैलकुलेट करने के लिए फॉर्मूला है 2 to the power n, where n is the degree of heterozygosity. Now see the case number 1. Capital A, small a. Here both alleles of one gene are different, so degree of heterozygosity is 1 and the value of 2 to the power 1 is equal to 2. So 2 types of gametes will be formed in this case. Now see the case number 2. Capital A, small a, capital B, small b. Here value of n is 2. As you can see, heterozygosity in alleles of 2 genes. So number of types of gametes formed will be 2 to the power 2, which is equal to 4. Similarly, you can calculate in case of 3, 8 types of gametes will be formed because value of n is 3. In case of 4, 16 types of gametes will be formed as value of n is 4. And in case of 5, again 4 types of gametes will be formed because here also heterozygosity is at 2 places and value of n is 2. Point 36. Number of meiotic and mitotic divisions take place for formation of an embryo sac from megaspore mother cell. 1 meiotic division and 3 mitotic divisions are required for formation of an embryo sac with 8 nuclei and 7 cells from megaspore mother cell. 37. Number of meiotic divisions required for formation of 1000 seeds. 1250 meiotic divisions are required because here 1000 meiotic divisions are required to get 1000 female gametophyte or embryo sacs and 250 meiotic divisions are required to get 1000 pollen grains. Now after this, add the two values 1000 and 250, answer will be 1250 meiotic divisions. 38. The world population in 1900 and in 2000 were, it was 2 billion or 2000 million in 1900 and 6 billion in 2000. As per census 2001 report, the population growth rate was, it was 1.7%, that is 17 per 1000 per year, which is the rate at which our population could double in 33 years. Point 39. In humans, the embryo's heart is formed after 1 month of pregnancy. The first movement of fetus usually observed during 5th month of pregnancy. Eyelashes are formed by the end of 24 weeks. Fetus developed limbs and digits by the end of 2nd month, and most of the major organs are formed in fetus by the end of 12 weeks. 40. Mendel's monohybrid cross is mathematically condensable to the form of binomial expression. This expression is Mendel's monohybrid cross represented F2 generation to be in the genotypic ratio 1:2:1, that is capital T, capital T, capital T, small t and small t, small t. This is condensable in the form of binomial expression ax plus by whole square. Here x is equal to capital T and y is equal to small t. Also, gametes bearing genes capital T and small t are in equal frequency, that is 1/2. Half of the gametes will be with capital T and half will be with small t. So, A and B की value है 1/2 and x and y are capital T and small t respectively. Now put the values in the formula. It will be 1/2 capital T plus 1/2 small t whole square, and the value will come out to be 1:2:1, the ratio of monohybrid Mendel's cross. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.